All right, good morning. Is on there. There he goes. So I'm going to take a slightly different track to Stephen yesterday on the internal conflict in cancer, and I'll tell you. I'm just going to draw a quick cartoon and tell you why. So I'm an oncologist and mathematician, and so I sort of work in between the two in a group we call the theory division, and I'm going to give you a few slides at the very beginning to introduce the group over at large. But there's a couple different ways you can look at internal conflicts in cancer, if you will, and I'm going to use red and blue. So we could have a host, like hopefully none of us, with a tumor inside them. And that could be a conflict between the tumor and the host. But you could also have within that tumor, you could have a tumor and have internal conflict within that. So you could have some types of cells and some other types of cells in conflict or cooperation. And it's actually the latter that we're going to talk about today, trying to think about what's going on within a tumor, if there are conflicts, if we can measure them and point them out, and then if we could take advantage of them. So the idea for the talk today is harnessing those internal conflicts for a novel idea behind therapy. So accept that there is a tumor, accept that I'm going to hopefully prove to you there are conflicts and cooperations. Is cooperation a conflict? I don't know. Interactions, negative interactions, yeah. So that's what we're going to talk about today. I'm going to go through a couple of philosophy stuff at the beginning just to give you a sense of what we do more broadly, because I think this is a really welcoming community and there's a ton of really interesting overlap. And thanks to Arvid and Manis and everyone for organizing and inviting and letting me be here. So yes, okay, so I think I'm an oncologist, but it's important to remember, also as Dr. Frank said yesterday, microbes and cancer are very similar in many ways. And really, they are, I think, the two evolving diseases, which are the most important part of the next generation of biomedicine. The diseases that actually evolve, because we have these incredible medicines that we make, but the evolutionary process is smarter than them. And so the Red Queen hypothesis is writ large in biomedicine all the time. But that said, it's not doom and gloom. We cure most infections with either just waiting, because the immune system's an amazing co-evolving system, or with radiation. So I do a little advertisement for our field, but also with a knife. Chemotherapies do not cure most patients, sadly, but and we've probably all seen some pictures about failures of those therapies. Almost all the time when a cancer becomes metastatic, it means it's spread from its normal position and it's no longer extirpatable. You can no longer remove it. It's chemotherapy or systemic therapies that are really the watchword. And for the most part, patients are not curable by those. And again, it's because these tumors evolve. And so in this picture, this is a classic picture lots of folks show in these settings of a patient with melanoma who has a very specific, this is a lucky patient who has a very specific mutation that has a drug targeted directly to it, a targeted therapy. After about 4 weeks of therapy or 16 weeks of therapy, it goes away, but not entirely, because it turns out that 0.1% of 10 to the 13th is still a big number, and it roars back. And I think that this is the classic idea. We give the medicine on the left. We have a cartoon of a tumor with some rare red mutants. We give a medicine that kills off the gray ones that are sensitive, and the red grows out. I'm going to show you a concept today about something called adaptive therapy, which is trying to not kill off all the sensitive ones and instead take advantage of a potential conflict within the tumor by modulating the frequencies. It's very similar to the idea of theoretical ecology and modulating wolf and moose populations, for example, in a game reserve, where maybe the moose population is affected by the predation. Of course it is, right? So if you mess with the wolves, things happen to the moose. And if you can model and specifically put numbers on those parameters, you can actually move toward a control theoretic framework. And that's sort of where my lab wants to go, writ large. But I do experience a whole bunch of cognitive dissonance in my life. On the Fridays when I take care of patients with cancer, all of these sort of beautiful models fall apart. I don't really care. They don't really care. But when as a scientist, I really think about this process of starting with a single clone, this gorgeous exploration of state space that we talked about yesterday during the tutorial, which is amazing, and really setting up what we see as a tumor, which is a complex tissue that really is evolving and living within the host. These things take months and years. And by the time we stick a needle into a patient, it's all set up. It's already this, I'd say, beautiful, complex tissue. And so thinking about that process is what we do in the lab in a bunch of different timescales. And I think the idea of timescales is really central to what we do as well. So I'm going to give you 3 different timescales that we think about just as an idea and decorate those with some of the model types that we use. And then I'll dive into the proving to you that there's conflicts that we can get a hold of. So we call ourselves the theory division. I think I saw someone with a Joy Division shirt on yesterday. Yes, we stole our logo from them. But as a physician, or sorry, as a physicist by original training, I'm happily not married to tools, but I think married to questions. And well, I'm married to my wife, who's actually here in Chicago, which is why I didn't join you for dinner. But I think it's the questions that really drive me. And I think with analytical tools that are better and better and better, it's going to be us driving the questions that are more and more important. I think the scientist with the sharpest questions is going to win the day because the analysis is falling. We've written ourselves out of that one a bit. Okay, but we do a lot of stuff in the lab, going from antimicrobial resistance. So our most recent papers are actually about a specific kind of Staph aureus and some drugs that are of interest, but also the same questions in oncology. So how do evolution and ecology drive these drug failures, and what can we do about it? So again, this sort of overarching thesis is that cancers and infections are complex adaptive systems that change multiple timescales. And each of those timescales requires a different strategy when it comes to modeling, but also when it comes to intervention. And so I'm going to tell you about these 3 timescales kind of in a row, and this means that probably everyone here will kind of get this already. I'm going to name them just for convenience. I'm going to call the longest or slowest timescale an evolutionary timescale. Allow me that. And this is where mutation dominates. So this is sort of where we think about PopGen maybe more classically. And I'm standing in a room full of population geneticists, and I'm not one, so I apologize if that's wrong. But in this world, our lab thinks about ideas like fitness landscapes, fitness seascapes, and we think about how to maybe bias selection in some way. So I accept the fact that Darwinian evolution is going to beat my drug. Can I choose how it does? Or can I predict in what ways it might, such that I have a strategy ready for that eventual inevitable failure? And so we think a lot about those things, and here are some students who've done some of that work with us and some papers you can look at there. And then here, I'm going to call this the middle or clinical, or sometimes I call this the ecological timescale. And this is where we're going to focus most of today after I get through the beginning. And this is some clever students that are working real time. Jeff Maltus actually just started as faculty at University of Maryland. Very clever undergrad biophysicist, now working in EEB. He's actually formerly in biology, but thinking really hard about some of these questions. And then Artem in the middle is now faculty in Utrecht, a very clever computer scientist who's working on what he calls algorithmic biology. And then you see some other folks, Mina and Nathan. But really, this timescale is, I'd say, faster. And this is where we think about more theoretical ecology type approaches. So this is where we have the wolves and the moose, for example, like a theoretical ecology framework. You might not even need mutation for you don't need mutation for population fluctuations. Maybe you need a harsh winter and all of a sudden populations fluctuate wildly. Maybe as an oncologist, I can impose a harsh winter type of idea, right? So can I take the tools from theoretical ecology, and again, there's probably theoretical ecologists in the room, can I take those tools and put them to use in anger to take care of the patients that we have in front of us? And then finally, there's this the shortest timescale, which is the now question. So after I stick a needle into a patient and I send it off for sequencing or whatever analysis I choose, what can I say about what to do today, regardless of what's going to happen tomorrow or the next day? And we spend a lot of time thinking about sort of genomic signatures of therapy choice. We're not going to talk about that today because I think it's it's the least relevant topic. But I think even evolution guides our thinking here because we have this genotype-phenotype mapping. Because what we usually measure in clinic, what we usually measure when we send a sample of your tumor out to some company to bring it back, we measure the genotype writ large, right? It's usually, like we said yesterday, it's a gemisch of all of the genotypes in there, but we get a sample back that is a genotype. And what we really want as someone who puts selection onto things is a measure of phenotype. So I think what we've been trying to do in the lab is come up with these genotype-phenotype maps to map from whatever I see today in the genome to a what to do question. So is it flying or is it scurrying? Should I smash it or should I shoot it? Kind of question. Okay, and we think a lot about those things. So that's the end of the beginning. So I'm going to reframe now the rest of the talk around this ecological or clinical timescale. So this is fast, where mutation does not matter. Maybe it probably still matters, but it might not have time to interact. And what I think's really interesting about cancer in particular is that it's one of the few places where these timescales get shoved together, because a lot of times you can ignore these ecological types effects in bacteria because it goes so fast. Well-mixed experiments are probably just as good as spatially derived ones. Don't shoot me for saying that. Biofilms, yes, there's lots of reasons why it's maybe not perfect. But in general, virus, microbe, you can think about, and mutations happen fast. So mutation is probably enough. And you don't have to worry about the ecology part necessarily. And like I said, in wolves and moose, you can kind of ignore the mutation. It doesn't really matter on the timescales of interest. Cancer is this fun place where, because it's not really like a regular life form, mutations going fast enough and ecology is happening at the same or close to the same speed. So I really do think in oncology, we need to think about how to shove those models and timescales together. So it's like I gave a talk once called Twisted Timescales. I really think this is where things get messy when you can no longer put an epsilon in front of something and make it go away. So I think that we need to try to think about mathematically, and I'm going to show you just a preprint and a title you can go read at the end of how we're thinking about putting those two formalisms together, which I think will be important. Okay, so this idea that we can model tumors like an ecology, what kind of evidence do we have there? So I think people have been thinking about this for a while. I'm not the one who invented this idea, nor even was my supervisor, though I stole this slide from him. I will say that when I started my residency training, so my specialty oncology training, which is 2009, which is almost 20 years ago. That's disturbing. It actually was quite common for oncologists, especially practicing oncologists, who, sometimes by the time you're in practice 20 or 30 years, you're not really in the newest biological textbooks anymore. Fair enough, you're practicing. And so I think even when I started recently, because I'm quite young, right? It was not uncommon for oncologists to think of tumors as balls of mutated cells that were all kind of the same. As a matter of fact, I have a very specific memory of one of my first days on the breast cancer service, and a woman came back with recurrent disease 8 years later. Sad situation. It was good. It ended well. She had a metastasis in a bone. And I said to my attending physician, I was like, well, shouldn't we probably biopsy the recurrence and see what it is? And she said, well, why? We know what it is. We have her biopsy from 8 years ago. And that was, and still is in many cases, what you do. You don't think about the intervening years as something could have fomented great change, right? Clearly, however, that's changing in the oncology, in the cancer research community. That's now obvious that things would change, and you'd want a biopsy again. And I would argue in most clinical scenarios as well. So it's not even just that the tumor cells themselves are changing over time and becoming more interesting and heterogeneous, but they're also, like we said yesterday, recruiting in blood vessels, recruiting in normal tissues, because now they are much more than a collection of mutated cells. They are a self-sustaining little civilization with all the trappings, most of the trappings of other complex tissues. And so that sets up a problem, right? Because they can regenerate, they can grow in, they can feed themselves, but they can evade the immune system, they can chew up drugs in different ways, they can cooperate. But it also sets up possibly an opportunity. So if you want to roll back the clock to whatever date that says in the upper right, 2003, this is the first paper from Bob Gatenby, who was a great friend and mentor of mine, who actually encouraged me to do my PhD during residency. And this is one of the very first applications of evolutionary game theory to oncology. And you can see a sort of a game theory model on the left, thinking about normal versus cancer. So it's more like this problem here, right? But then as we're moving forward, what we're thinking about in the bottom left, this is actually a paper from a friend of mine called David Basanta, whose picture will show up next. It was really the first time he moved into this scenario. So can we look at two different types? And in this case, in the bottom left, you see a payoff matrix, which I don't want to belabor, but if you don't know what that is, it's a matrix of parameters that you shove into a series of dynamical equations that I'll show you in a minute, but I don't want to focus on. But this is really the first time he moved toward the idea that you could model the internal civilization of the tumor itself rather than the host. And what's important, I think, is that, and this is my gateway drug paper. You can see my name in the middle there somewhere. This is the first mathematical paper that I participated in, and it was a game theory one. And this sort of started an entire saga that led down the road of becoming a scientist. Woe is me. No, the rejections just began there. And so what we studied here was a game theory model of a cancer with 3 types of things. I'm not even going to tell you what they are because it doesn't matter. S-type cells, D-type cells, and I-type cells. And what we did was we felt quite clever. We wrote down this payoff matrix in the upper left. Then we could write down, in this case, discrete time replicator dynamics. And we could model how a tumor changed over time. And the point I want to make with these pictures is that you could start asking questions that most of the time you couldn't as a physician. So we could ask, what happens if you intervene early compared to late? So you start the simulation running, same exact simulation, same parameters, and then at some point when the blue line turns on the left, you turn on a therapy of some kind, all of a sudden you see the dynamics change, a new steady state is reached, and now green wins instead of blue. If you do that same therapy a bit later, you still stay with blue, right? So early intervention mattered. You could also ask how long a duration of therapy might evolutionarily affect a tumor, I should say, ecologically affect a tumor. And again, here you see the same therapy given at a short, medium, or long impulse gives you a different long-term steady state. Now, this is relatively obvious to someone who studies dynamical systems. There are more than one attractor states. You change things, it moves to another place. However, it was not that obvious to oncology that you could think about it in this way. So all of a sudden, you can ask questions like when to treat, how long to treat. But of course, the mathematicians in the room are saying, well, it's all relatively obvious once you've written down that. True. That didn't stop us from writing another theory paper about the same thing. So similar model system, you'll notice is a slightly different formalism, but the idea is the same. And this is from Artem, the student that I told you is now in Utrecht, thinking about similar things. In what order do medicines matter? So if I give one drug and then another, A and then B and then C, is that the same thing as giving A and then C and then B? An evolutionary biologist, to them, it might be obvious that exposing a creature to winter and then summer is different than summer and then hotter summer, for example, or in a different order, right? If you evolve in one scenario and then climate changes to another versus the opposite. But again, this depends entirely on what you've written down. These are deterministic equations. There's nothing here that's magical. And a big question too is like, sort of, does this matter? Are we just telling stories so far? And I think to this point we were telling stories, important stories that help us reason and think about tumors overall, but not ones that we could do something about. But it turns out actually these matter. So if you take these ideas and you move forward toward a clinical trial, which seems audacious, but was done at Moffitt Cancer Center, you can take this idea of something called competitive release, which is the idea that a sensitive ancestor can outcompete a resistant clone that has arisen from that sensitive ancestor, given no drug situation. So in the absence of therapy, the sensitive can actually beat down the resistant. Once the therapy turns on, we saw the resistant grows out and the patient's in trouble. This clinical trial took advantage of that and it asked, could it modulate the turning on and turning off of drug in such a way that you were basically promoting the sensitive in certain scenarios? So could you promote the sensitive, like in this lower right-hand side, you withdraw the drug, the sensitive comes back and outcompetes the resistant ones that are going to kill your patient. And then a little while longer, you put the therapy back on when the tumor's grown, and you sort of modulate it over time. And they're calling this an adaptive therapy approach. And it worked in this clinical trial. There's now a series of other ones. But I think it matters. And I'm just making sure I was 9:10 to 9:30, right? Okay, we should be good. I'm going to go a little bit quickly through some of the data just to get you there. But I think that the story is the more important part anyway. But again, it mattered. It worked. But it really depended on what was written down in that matrix, right? Because if you don't know what's there, you can't model the system and you're in trouble. So what we asked was, can we get our hands on those parameters? Can we measure those in some way? So we designed an assay for it, and I'm going to show you, I'm going to rip quickly through what that assay showed, just because I want to get to the concept. But the idea is, how can I design an assay designed specifically to measure the parameters of that mathematical model and measure the game being played, if you will. So we took a series of cells that you can buy from the store that are sensitive to a drug called alectinib, ALEC-S, and we exposed them to that drug over a course of 6 months. And Darwin was right. They all became resistant. So now we had a red mutant and a green ancestor, and they came from the same place, so they had some relatedness. So we could all, there's probably some inclusive fitness arguments we could make here. This is why I got excited yesterday. And we asked the question, can we measure, or is there any ecology or cooperation or conflict happening? So we ripped down the dynamical system that is the replicator dynamic into its simplest form. So this is a two-player replicator dynamic. It's a first-order linear ODE. And you can see what we're looking for is we're just looking for those A's, B's, C's, and D's. Those are what populate that matrix that we did all that maths with earlier. So the simple question here is, can we design an experiment such that we can get A, B, C, and D? So if you mix these cells together, this is red and the green. If you put a whole bunch of the drug in, it turns out that the sensitive ones die and the resistant ones grow out. That's not surprising. But what we want is the dynamics over time. We don't want one went up, one went down. And so what we're going to do, I'm going to build this chart for you, and I think what's going to pop out is, yes, there's cooperation. And everyone's going to say ooh and ah, and then I'm going to be done with my time. So what we're going to look at here is a complex picture, and I'm going to build it up one spot at a time. So on the x-axis here, you have time. On the y-axis, you have cell number, raw cell number of each type. And you see on the bottom, there is drug, there's some dose of the alectinib, and there's no cancer-associated fibroblasts. So call it winter, if you will. There's another scenario where this is the same thing. So what we do is we actually put those together and count them in time. It's actually quite nice, nicely linear on a log scale, pretty neat. So it fits an exponential pretty well. But we can also start now taking those exponential fits and put them into a larger major axis. So now I have growth rate of the resistant type on the y, and I have growth rate of the ancestor on the x. And so you can see we're here in the upper left quadrant where the resistance growing and the parental's dying because there's drug. In a different scenario where there's no drug, both are growing. All of a sudden, if we take all four scenarios, winter and summer, drug and no drug, we can see this large shift. And so what you see within these convex hulls is the expected evolution of how therapy moves. So you add therapy, you kill the sensitive ones, but you maintain the resistant. And so what we see here is pretty neat. So let's take one of those convex hulls, just this one, just the blue one, and break it out. And what we're seeing is plots similar to what we saw yesterday, some of these. So this is parental proportion on the x. And what we're seeing is the growth rates of each changing based on the presence, the frequency of the other. And what we see is frequency dependence. And so this is a signal of, in this case, I'd say a conflict or a cooperation, however you like to think of it. But what we also have is the opportunity now to pick those parameters off the plot, because we can look at our intercepts and really pull those parameters. So I think what's really cool about this is that we now have the ability to nail those matrices. And once we do that, we can do all the math we had before. So we have a payoff matrix for a scenario, a single scenario in cancer. And this really gives us an idea that these fitness functions in game space real qualitative shifts. So you can sort of think about what's the evolutionary or ecological shift that adding a drug does, and what's the evolutionary ecological shift of winter and summer. And so thinking about those things in a different way. Can we cast these problems in terms of these scenarios in order to think about therapy differently? So can we harness these conflicts? What's cool is that we did it again. I'm just going to skip this slide, but the point is that if you do this again with different drugs and different cells and different doses, your games move all over the place. So we actually have access in our armamentarium to drugs that perturb the scenario in different ways that you can choose. And so we actually have now a whole library of scenarios and drugs where we can pick the game. So all of a sudden you can start stacking up the dynamics in a way that you like. And that's sort of where we want to get. I want to get to a place where we can sort of start applying formal control methods to these ideas. So we can think of those in the model systems that we use. And that's the end. So I just wanted to say that I think that we've sort of shown that evolution drives drug resistance. We knew that partially through measurable ecological interactions. You can read that there in Artem's paper. Sorry, I did this wrong. And then I think we have an in vitro assay that works pretty well. There's another paper in a different kind of lung cancer. And then I think I was going to present, I was torn. This PRX Life paper just came out last year. I think it might be really interesting to this audience. So look up Maltus et al. But I think the in vitro assay is really begging us to come measure what this evolutionary phenomenon looks like across a lot of scenarios. And then I think the last one is this preprint that I'd love folks to see, as I think we've taken this ecological dynamics idea and layered mutation on top. Because even though I said it's slow versus fast, cancer really squishes them. And so I think that we're ignoring mutation, but it's clearly important. And that's what I wanted to say. So I think it's just our ability to harness the conflicts presents a new opportunity for intervention. I want to say thanks, and I'm done.